Welcome friends, myself Shoptaporni from Biolife. Today we will continue our previous lesson on apoptosis. Okay, so let's begin. Okay, first of all, today we will learn about caspases. What are caspases? Caspases are family of proteases. Proteases means those proteins which can degrade other proteins. Okay. So, all caspases have a cysteine residue at their active site. That is why this C has come from the cysteine residue at their active site. Okay. Now, another thing is that all caspases cleave other proteins at one or two specific aspartic acid residues of their target proteins. That is why this ASP has come from the aspartic acid residue. Okay. Now, all caspases are formed in our body in an inactive form known as procaspases. So, procaspases are the inactive form of caspase in our body. Okay. Now, to activate this procaspase into an active caspase, what do we need? See, procaspases actually look like this. Okay. Think this is the cleavage site. So at this side obviously aspartic acid residue will be present, and at this side this procaspase will be cleaved by another active caspase. By the proteolytic cleavage of this procaspase, it will form a heterodimer like this. Okay. See, a heterodimer like this. From this point, it will be cleaved and form a heterodimer, HD. Now, when a heterodimer will join with another heterodimer, like this, it will form a tetramer. See, this is the tetramer, okay? And this tetramer is nothing but the active caspase of our body. So, this tetramer is the active caspase. Okay. That is how inactive procaspase is converted into an active caspase. Now, one question may arise in your mind that how does the initiator procaspase is cleaved and get converted into a initiator caspase? Because at that time, there is no active caspase like that when the initiator procaspase will be cleaved. So, how does that happen? Let's see. Initiator procaspase looks somewhat different than other procaspases, okay? Because it has a prodomain. This is the prodomain of any initiator procaspase. And this prodomain contains a card or caspase activation recruitment domain. So this is the initiator, initiator procaspase. Okay. Now, this card domain, when interacts with an activator, when interacts with an adapted protein, it helps the procaspase to come close with another initiator procaspase, okay, and form an activation complex like this. So, this card domain along with the activator adapted domain, adapted protein helps to procaspases to come in close contact. So now two initiator procaspases are in close contact, and now these two procaspases will cleave each other. Okay, this one will cleave this one at this side, and it will also cleave this one at this side. Similarly, here cleavage will also occur in vice versa manner. And as a result, these two initiator procaspases will form the initiator caspase, okay, like this. See, this is the initiator caspase which is now active, and prodomains will be cleaved with their card domains, okay. So now this initiator caspase will activate other executioner procaspases like the way I have already told, and will form the executioner caspase. So these initiator procaspase will now cleave executioner procaspase. Let's write PC. PC means procaspase, and will form executioner caspase or executioner C. Okay. Now this executioner C, which is active, it may either cleave other executioner procaspase and form executioner caspase, or it may directly cleave other target proteins of the cell, of the apoptotic cell. Okay. Now that is how the Caspase cascade starts. This is the Caspase cascade. And what are the target proteins? Target proteins are number 1, nuclear lamins. Okay. That means when executioner procaspase will cleave the nuclear lamins, the nuclear membrane will be distorted. Then it will cleave inhibitor protein of endonucleases. Okay. We know that endonucleases cleave the DNA during apoptosis. But in a normal cell, those endonucleases are inhibited by other proteins. So to activate the endonuclease, you need to inactivate those inhibitor proteins. So the executioner caspase cleaves those inactivated proteins and activates the endonuclease so that they could break the DNA. Now, executioner caspases also cleave cell adhesion materials or molecules, okay, cell adhesion molecules, so that an apoptotic cell can detach from other neighboring cells and can form a round-like structure. The round-like structure is easy to be engulfed by other macrophages, and it will also degrade or cleave the cytoskeletal proteins, okay. Because if you can distort the cytoskeletal protein, the cell will automatically be smaller in size and it will round up. Okay. So that is how apoptosis begins, and this is the Caspase cascade of the apoptosis. So from this it is evident that Caspase cascade is destructive, obviously destructive. Second thing is that it is self-amplifying, because you have already seen that whenever a executioner caspase is activated, it can activate a chain of executioner caspases and can spread the death signal all over the cell. So obviously it is self-amplifying. And the most important thing is it is irreversible. Okay, that means whenever an apoptotic signal comes, if the initiator procaspase is activated, nothing can then stop this pathway. Okay, so this pathway is irreversible. So that is all about the caspases. And another thing is that not all caspases are always required for apoptosis. Some caspases are functions in inflammatory process. Okay, so inflammatory caspases are mainly caspases 1, which is also known as ICE or interleukin 1 converting enzyme, which converts the precursor of IL-1 to its active form. Other inflammatory procaspases are caspase 4 and 5. Now the apoptotic caspases are of two types, okay? Either they could be of initiator apoptotic caspase or they could be executioner apoptotic caspase. Initiator apoptotic caspases are caspase 2, 8, 9 and 10, whereas executioner caspases are caspase 3, 6 and 7, okay? So, now let's discuss about the pathways of apoptosis. There are two pathways of apoptosis in our body. The first one is extrinsic pathway and the second one is intrinsic pathway. What happens when any kind of bacteria or virus attacks our cell, then that cell is needed to be apoptosized, okay? Then the death signal that this cell has to be apoptosized comes from the outside of the cell, okay? That is why this kind of signal is known as extrinsic signal and or extracellular signal, and this type of pathway of apoptosis is called the extrinsic pathway. So in extrinsic pathway, what happens? Let's say this is our target cell, okay? This is our target cell and this is the cell membrane. In our target cell, this is the target cell which will be apoptosized, okay? So, on the cell membrane of the target cell, these are the death receptors, okay? Death receptors are present. They are homotrimer. Death receptors have three domains: an extracellular receptor binding domain, then a transmembrane domain, and then a cytosolic death domain. This domain is known as death domain. This red colored cytosolic or intracellular domain is known as death domain or DD. Okay. Now, in this case, this is the receptor of Fas ligand. That is why we will write its Fas receptor. Okay. Whenever a killer cell like this is a killer lymphocyte. A killer cell like this brings the death signal in a form of Fas ligand. Okay, these are the Fas ligands, and these Fas ligands, when binds to these cells, this Fas receptor, what happens? Then, as a Fas ligand now binding to the Fas receptor, it activates adapter proteins inside the target cell. Okay, adapter proteins look like this. See, this is a adapter protein. Think this is a adapter protein. So adapter protein also have a death domain. This is the death domain. Okay. So two death domains bind with each other, and this adapter protein, this is the death domain, and it is the death effector domain or DED. Okay. And this adapter protein is known as FAD, FADD. That means Fas activated death domain. Now, when the adapter protein binds with the death domain of the receptor, Fas receptor, then it brings the procaspases, initiator procaspase, mainly the procaspase 8 and procaspase 10 to bind with this adapter protein. Okay. So, think this is the procaspase of our body. It could be procaspase 8 or 10, okay? And we have already discussed, it is its card domain or also known as death effector domain, DED or card domain, okay? So, this DED or card domain, what happens? It binds to the DED or death effector domain of the adapter protein like this. Okay. Similarly, also adapter protein binds, and with that another procaspase binds here, okay, and they come to a close proximity and forms a signaling complex. This signaling complex is known as DISC. DISC means death inducing signaling complex, okay. So now these two initiator procaspases cleave each other and form the initiator caspase, okay, like this. This is the initiator caspase. It could be initiator caspase 8 or caspase 10. So let's write C8 or C10. Now this initiator caspase activates other executioner procaspase to form executioner caspase, and then they starts the caspase cascade and breaks the target proteins, okay. And that is how the cell gets apoptosized and shrinks, okay, and then the macrophages come and engulf the cell. So this is the total pathway of the extrinsic pathway of apoptosis. This is the extrinsic pathway of apoptosis. Now see, not always the death signal, whenever the death signal will come, our cell must be apoptosized. That is not the factor, okay. Sometimes inappropriate death signals also come, and at that time our cells, target cell must not be apoptosized. So how that will be inhibited? So for that inappropriate apoptosis, there are two inhibitor proteins in our target cell. That is, one is decoy receptor and the second one is flip, okay. What happens? See, this decoy receptor mimics the FAS receptor, but it lacks the death domain. So what happens? Those decoy receptors are also present on the cell membrane, but whenever the FAS ligands binds to them, they can bind to that ligand but cannot induce the apoptosis because they lack this death domain, okay. So they competitively inhibit the inappropriate apoptosis. Now another one is flip. Flip resembles the procaspases, okay, but they also lack the proteolytic domain of the procaspases. So whenever the death signal comes, this flip competitively binds to the adapter protein, okay, and it competes with the procaspases, but it cannot induce apoptosis because it lacks the proteolytic domain. So it also helps to prevent the inappropriate apoptosis in our cells, okay. So this is all about the extrinsic pathway of apoptosis in our body. Okay, with this we end our today's lecture here. We will continue this chapter and we will learn about the intrinsic pathway of apoptosis in my next video. So please like, share and subscribe to my channel to stay updated. Thanks for watching and happy learning.